welcome back to another late night recording, lecture 21. So getting pretty close to the end of the semester. Hope you guys have recovered from the exam. Life isn't too stressful. You have plenty of coffee. The image on the screen really has nothing neuroscience about it, but my guess is that as you look at it, it appears to be moving. And I promise there's no animation, there's nothing moving on the screen. It's one of a fairly large class of visual illusions that use odd features of how we process light and dark, edges, that kind of stuff, to give the illusion of moving in the bits of the image you're not looking at a particular moment. In general, if you focus on any one thing, you can see it's not moving, but the bits that you aren't directly focusing on appear to be kind of writhing around. And our visual system is pretty easy to trick in this way. There are some really cool illusions, but exactly why they work, no one really knows. A couple things to kind of fill in gaps from previous work. So we spent a bunch of time talking about memory, and you probably recall one of the things we talked about was that the way LLTP, long-term potentiation, seems to put in place permanent, or at least very long-lasting synaptic changes, is by trapping nuclear proteins, sending proteins that are synthesized in the nucleus, at least partly as a consequence of activation of Krebs. Those then go throughout the cell and get trapped in what are tagged synapses, so the places where the engram is being changed. That tagging system, again, one of the many things that during the semester I'm going to tell you, we don't really know exactly how it works. One cool development, this is a couple of years old now, but one thing that was also discovered is there are some individual proteins that can last for a really long time. So in general, proteins in the body, in the brain, throughout your whole nervous system and elsewhere, turn over fairly fast. We're not set up so an individual molecule in general lasts for a particular long time. Things like AMPA channels, cycle in and out of the membrane. Any individual channel may get recycled pretty rapidly. There are some proteins that's not true. So what this particular study did was look at that class of proteins that have very long half-lives. They call them LLPs, long-lived proteins. And there are some places in the body where you find these in particular. I think the eyeball is one of them, so the lens of the eye. Some collagen structures, things like tendons, the bridge of your nose, that kind of stuff. There are certain places where proteins last for a really long time, years. What this study did was look to see, okay, is that a possible explanation for some of the things we see in synapses? Are there proteins at synapses that have such long lives? And so they looked at cytosolic fractions and synaptic fractions of different neurons. What they found was that, if you enrich the environment of these mice, and so if you cause them to have more memories, more strengthening of LTP, more connections being made, in fact, those synapses increase their concentration of long-lived proteins. And you can track this across time. The study identified well over 100, I think 164 long-lived proteins. And some of them, about 50 or so, had half-lives of more than a year. So this is an individual protein molecule that if you look across the span of a year, only has a 50-50 chance of being degraded at all. So very long-lasting, quite likely to be a way in which changes in a membrane, changes in a particular synapse, can hang out for a long time because those proteins are just going to be there for a long time. And so if they've caused a change in function of that synapse, it'll be persistent. A second way that some potentiation, some memories may be encoded, we looked previously at the protein ARC. I told you it stands for activity-related cytoskeletal protein. So it's a protein that's expressed when a cell has been undergoing plasticity. One of the cool things about that particular protein is that it may well be of viral origin. It may be something that the mammalian nervous system and then eventually the human nervous system kind of adopted and picked up from an ancient virus somewhere way in the midst of evolutionary history. If you don't have that protein, if you make an ARC knockout mouse, memory is almost absent. It's very hard to learn new things. It's very hard to form long-term memories. And it turns out that ARC has a structure very much like some of the viral capsules. Obviously we're all looking at coronavirus these days. What coronavirus does is encase itself in a protein membrane shell and go off to infect other cells. ARC may do something similar. And so ARC may actually have a role in allowing neurons to communicate and effectively infect one another. Exactly how? Guess what? We don't know. But the structure of this particular protein, the structure of ARC, is kind of cool and looks like it probably came from a virus. Looks like it may be, potentially even delivering bits of RNA back and forth between neurons to carry messages, to carry information, to say, hey, I'm feeling potentiated. You should be feeling potentiated. Can we strengthen this particular synapse? It's an active field of research. There's a lot of work going on, but it's kind of cool. It's not quite as cool as some of the popular press. So an ancient virus may be responsible for human consciousness. No, not so much. We're going to talk about consciousness a little bit from the next class, but ARC is certainly not the cause of it. And last time I was talking a little about memory fugues, states where you lose your memory for a few years. There are long-term situations where you lose consciousness and, as far as we can tell, make no memories for many years. This is a case where the return to consciousness was artificially created, so it was responsive to simulation. This is TMS, transcranial magnetic stimulation. We've seen this previously. And after more than 2 decades, this woman was moved to a new facility where they decided to try to intervene. This apparently persistent coma, almost vegetative state. Okay, what have we got to lose? Let's apply some TMS, some transcranial magnetic stimulation. It woke her up. And as far as we can tell, it's almost like the brain was rebooted. It came back to life. Much of the function appears to be intact. She can't walk very well. She couldn't walk very well before the coma. And so there is some hope that even in these long-term unconscious situations, maybe neurogenesis is still going on. And if you provide external stimulation, if you increase the chances those neurons can form connections and survive, perhaps you can repair whatever damage has happened sufficiently to allow consciousness to return. Certainly in this case, success. All right, that's the end of digressions. We were talking about memory last time, some instances of amnesia in the movies. I gave you Memento as an example of a movie that is really pretty relevant and pretty good in representing how people with amnesia, particularly Alzheimer's, can often use external recording devices, whether it's Post-its or tattoos, as a prop, as a way to increase their memory performance. And we spent a bunch of time thinking about false memories. In the Q&A section, yesterday, I guess, when I'm recording this, we spent a bunch of time talking about how a false memory is a poor terminology. It's a real memory of a thing that turns out to be factually untrue. But from the point of view of your brain, it's a perfectly real memory. Move from that into looking at memory modulation, the role of hormones and vagal stimulation and glucose supply and so on in creating an environment that says, hey, this is important. Basically, you're aroused, you've got some element of stress. This is something that should be forming a memory. It's going to be important to recall this. You're walking through the savannah, a tiger jumps out. Remember where the tiger was hiding. Or you find a particularly good source of fruit. Remember that where that fruit tree is. Anything that causes a signal of salience, signal of importance, is going to be something that makes that memory more important. Therefore, over evolutionary time again, we have got mechanisms that allow us to store memories for things that are tagged as important and not store memories, not waste the resources on storing memories for every random bush you walk past that don't turn out to be particularly interesting. No food, no tigers, no nothing. In the context of fear in particular, that emotional tag, that signal, hey, this is important, takes place in the basolateral amygdala. So you've got some aversive stimulus that says, ouch, or scary, or just annoyingly bright colors, whatever it is, something aversive gets paired with some of the other context, some other stimulus that you're experiencing at the same time. That pairing takes place in the BLA, causes a new memory in particular that has an emotional component. And so then the output of that through the central nucleus over to the hippocampus and so on is to say, hey, this particular memory, it's kind of important, hang on to this one. We talked about the fact there are other brain regions involved, frontal cortex is one, but several other places in the brain, as well as the amygdala, do deal with emotions. The basolateral amygdala is particularly good at dealing with anxiety and fear and so on. But you can see here, the orbital frontal cortex, some of the cingulate cortex, in fact, some of the insular too. We encountered this when we were looking at language. So injuries to this part of the brain, particularly on the left, tend to cause deficits in language production and so on. Probably there's a role for this part of the brain also in processing emotional states. The end product of all that processing is modulation And biochemically, that's essentially norepinephrine, right? The end product is to increase amygdala norepinephrine. That's caused by vagal stimulation, by peripheral epinephrine, and by an increase in glucose that both promotes hippocampal function and also increases amygdala norepinephrine. If we look at individual animals that are having an aversive memory caused, then their individual elevations in amygdala norepinephrine are directly correlated with how well they remember that particular aversive stimulus. Animals down here that show very little or no rise end up doing particularly poorly on the task. And there was a question, in the online discussion, why does too much stress not just keep making you better and better? Why is something that's really highly aversive, really highly emotional, not cause really, really good memory? Just like caffeine or glucose or pretty much any stimulant that you might think of, where a small dose increases memory performance, there's an inverted U curve. It's also called a Yerkes-Dodson curve. It's common across all this kind of stuff. And you can think of it as being very much like a signal to noise ratio effect. So if I have a little glucose or I get slightly on edge, that's going to increase the excitability of neurons. And the first thing that's going to happen is the neurons that want to fire, the neurons that would be involved in encoding a memory or indeed in retrieving a memory, they're now able to fire a little better than they would be otherwise. There's a little extra glucose around, they're a little more excitable, there's a little more epinephrine, all that stuff is going to increase the excitability of neurons. So the first thing that happens with little excitability is you get an increase in signal. And therefore the signal to noise ratio increases, you perform better. As you increase the amount of stimulant, caffeine, glucose, amphetamines, whatever it is, you can increase excitability a little further. And now also neurons that you don't really want to fire start firing. Neurons that don't code for grandma carrying axes, but code for grandma listening to Michael Bublé or wearing pink socks or who knows what. Things that aren't relevant. So an increase in noise. And so although the signal went up initially, now the noise is also going up and you end up getting no overall benefit, no increase in the signal to noise ratio. And in fact, too much stress, something like PTSD or chronic elevated glucocorticoids, effectively the noise goes up so much, it drowns out the signal. You actually end up getting a deficit in performance. You go below baseline. So too much stress, too chronic stress, bad. A whole bunch of consequences, right? So if you're just getting a little bit of short-term stress, you mobilize energy. You take some of your fuel reserves out of the liver, for instance, out of glycogen, put them into the bloodstream as glucose, you can perform better. If that happens chronically, that's diabetes. Constant high blood glucose is a really bad thing. You turn down all these short-term mechanisms. You might remember Sapolsky in the video saying, you're running for your life, this is no time to ovulate. And so things like digestion, growth, reproduction, those are all turned down. But again, if you chronically repress those, that's got negative consequences. Ulcers really shouldn't be here. Ulcers have been shown to be bacterial in origin. There's a guy at UVA, my alma mater, who won a Nobel Prize for showing, look, no, this isn't stress caused. In fact, things that go along with stress, poor diet, infection, not taking care of yourself, those may contribute, but actually stomach ulcers are bacterial in origin. Anyway, short-term stress reduces pain. This is why if you're shot on the battlefield, or in my own case, I was out running half marathon the other day and actually ran into a snow stake, stabbed me in the thigh. I looked down and like, oh, okay, doesn't hurt too much. Got home and blood is pouring out of my leg. I literally hadn't noticed because the stress induced by the fairly long run had caused analgesia. In the long term, chronically, that can turn down your ability to experience emotion, your ability to care about stuff. So, in general, short term, fairly moderate stress is beneficial. Long term, very high chronic stress, very bad. And you can see the consequences, right? Let's say you're bullied either a little bit as a child, which most kids are occasionally, or really frequently as a child. Your chance of committing suicide goes up markedly with frequent bullying. Your chance of having an anxiety disorder. All these things later on in life, really negative consequences, really major long-term life negative consequences as a result of chronic stress, even many years earlier. It's going to have long-term negative brain impacts. All of which is a fairly long build-up and fairly long recap of the last class. Insert your own questions here. I will note the attendance at Q&A sessions has definitely been dropping off. there is a correlation between the folks that are coming to those Q&A sessions and their performance on the midterm. So I suspect that's going to be true for the final as well. If you can, I do encourage you to come to class times. Apart from anything else, I enjoy seeing you and interacting, but I do think it's definitely beneficial to be there. So we've left memory behind. We've left modulation memory behind. We're going to spend a class or 2 classes talking about really bad things that can happen to the brain. And pictures like this one on the screen tend to cause people to wince a little. So this is what happens when you are driving along, you let your kid in the back seat play with your house keys, and all of a sudden you come to a really sudden stop, whether it's a crash or it's jamming on the brakes. You can see here, the keys have gone up into the jaw and also into the eye socket. In this particular case, the kid got really lucky. The keys went along the eyeball, didn't damage the optic nerve, and this kid turned out to be just fine. But it's a really scary X-ray to have to look at, right? It is not something you want to happen to your child. The brain is relatively vulnerable once it gets injured, which is kind of an odd thing to say. It's inside the skull, it's inside the blood-brain barrier. But it has very few defense mechanisms once you get past that. So tapeworms, for instance, another picture you probably don't want to think of as being associated with your brain. If you get an infection in the brain, your immune system doesn't have access, can't protect you. The blood-brain barrier prevents those antibodies from getting in. So if you somehow got past that blood-brain barrier with a parasite, like a worm, well, now it can wander around, it can lay eggs in the egg sacs. For whatever reason, these kind of parasites tend to like to go to the limbic system, into the amygdala, but you can get fairly widespread infections. Post-mortem, and post-mortem tends to happen quite rapidly in some cases, you take the brain out, it looks kind of like Swiss cheese. All of these little egg sacs and holes are caused by worms laying eggs or digging tunnels, literally chewing their way through the brain. At this point, in general, the class starts to kind of wince and not want to think about this stuff. Parasites are a big deal for the nervous system. We talked very briefly back about Toxoplasma gondii, this thing that comes from cat feces, invades the nervous system, causes personality changes. There are a wide variety of organisms that can get into and enjoy dwelling in different parts of the brain. This Hawaiian snail is a good example. So you learn to surf, you're on a Hawaiian beach, one of these snails gets into your nervous system, gets into your mouth or your nose or into your eyes, and will happily take up residence in the lining of your brain, moving around the outside. That feels weird. It can actually be perceived occasionally, not very often, but occasionally, and obviously causes inflammation and so on. In some cases, these worms move into the brain, right? So generally it's okay. Generally your body can cope, but sometimes you get an infection that moves into the nervous system. That can be really nasty. The inflammation is pretty bad, and you get some of these weird cases where this is a series of MRIs, and you can see this kind of movement of this particular white blob. That particular white blob is a tapeworm. It's a tapeworm that was eventually removed from this guy's brain, and the way they removed it is they took repeated MRIs, waited for the tapeworm to come near the guy's nose, stuck a pick up his nose, and pulled the tapeworm out of his brain. This was 2 meters long. How did this get into his brain, you might ask? Tell us, Omar. So this guy had had some puffiness around his eyes, and he'd been in China at the time. He was recommended to use a poultice, put it on his eyes, made from raw frog meat. The raw frog meat turned out to be infected with tapeworms. And so he plastered this stuff over his eyes, tapeworms crawled into his eyeballs, crawled up into his brain, yada, yada. Parasites are great and scary, and some of my favorite things in neuroscience. I'll show you a video next time from Ed Yong, who just is one of the world's best science writers and loves writing about parasites. Really nice guy. In any case, once you get into the brain, there's very little you can do except physically remove the parasite eventually. It's hard to get drugs in there. The blood-brain barrier protects you, but also prevents things like drug access. Here's another example. This is one of the most recent that was written up. This is from the New England Journal of Medicine, so about a year ago. And again, all of these what look like kind of light dots, those are holes in the brain caused by a tapeworm chewing it up. This is not that uncommon. Be careful, wash your hands, not just because of coronavirus, but also because you really don't want to get a tapeworm up your nose. Not a good idea. I mentioned that one of the problems is meningitis. Meningitis is an inflammation of the meninges. We'll come back to that in a second. it causes symptoms often that are very similar to brain cancer. So the problem with brain cancer in many cases is not that the cancer itself would cause problems if it were on your hand or something. It's because the skull is very rigid. And so because the skull is very rigid and space inside is constrained, if you get a very large tumor growth, as you can see in this example, it squashes the rest of the brain. It's got nowhere to go outwards. It has to grow inwards. It will cause compression damage. So it will cause loss of function in the cells around the tumor site because they're being physically squished. And so you can see the lateral ventricle on this side looks like pretty normal. The ventricular system on the side with the tumor has been squashed almost to nothingness. There's a herniation, a movement of tissue across the midline. Basically, this half of the brain's all been shoved over and squished up. Another thing that also happens is that brain tumors in general, brain tumors in particular, tend to wrap themselves around blood vessels. So this is a PET scan of glucose use. Tumors have very high metabolic demand, and so they will tend to associate themselves with blood vessels, often, as you can see here, very deep within the brain. That makes removal really hard because, first of all, getting access somewhere this deep in the brain is really tricky. And second, the number of blood vessels involved, you really don't want to cause bleeding in the brain. That's a stroke. We'll get to that in a couple of slides. And so there are limited tools for being able to remove deep brain tumors. A surface brain tumor, not a really big deal. And in fact, often these will get detected almost by accident. So this chap had a bike accident, fell off his bike, went to his local ER for an almost precautionary CAT scan. And what they told him was, yeah, no damage from the bike accident. By the way, you know you got brain cancer? So he turned out to have a small tumor on the surface of his brain. And so what you can see here is him being prepped for surgery. This is the stereotactic frame we've encountered a couple times before. And he decided to document it. If you ever have brain surgery, you'll be awake for the process because you have no pain sensors, right, in your brain. 1 second, I'm going to sneeze. I'm going to pause the recording. Okay, I'm back. You really don't need to have me sneeze on cause your stress levels to go up too much. Anyway, there are pain receptors, of course, on the skull. If I tap you on the skull, you can feel it in the skin. But inside the skull, there are no pain receptors. So if you're having brain surgery, other than a topical anesthetic to reduce pain on the skin, you'll be awake. The surgeon will want to know what's happening. So this guy decided he was going to record his own brain surgery. So here you can see the scalp has been retracted, a layer of skin with a hair attached, being pulled back, exposing the surface of the skull. Now a piece of the skull has been cut away, and that'll be put back later on, exposing the dura. And if you peel away the dura, now you can see the brain. So you can see quite clearly here the vascular supply, the sulci and gyri of the brain. And here, what looks like almost a piece of fat or gristle, that is the brain tumor. It's a surface brain tumor. It's going to be cut out. This is actually a very, very simple brain surgery with almost no risk. You don't need to go deep and invasive into the brain. You just literally almost scoop it out with a spatula. And you get to take it home as a souvenir. So here's this guy's tumor. It's about 3.5 centimeters long. Little clump of tissue on the surface of the brain. Sew him up, put the scar under the hairline. Obviously, this guy shaved his head in preparation, but eventually if he grows his hair back, you'll never be able to know that there was anything wrong with his brain at all. And to be fair, there really isn't, right? The tumor was small, it didn't cause any problems, away it goes. More common, sadly, are tumors like this. So one thing that's also true about brain tumors is they tend not to be based on neurons. Neurons don't mostly divide. There's very little adult neurogenesis, there's very little division of cells. And so it's actually quite hard to make a neuron oncogenic, to make a neuron cancer. More likely, it's going to be what's called a glioblastoma. So a cancer of the glial cells, and commonly, as you see here, wrapped around a major blood vessel. Because the brain doesn't have an immunological system, sterility is really important. So when you're doing brain surgery, when we're doing brain surgery on rats in the lab, it's not quite to this extent, but you really have a very sterile environment. How do you get to that tumor if it's deep in the brain? You can't, in general, use a physical knife. Instead, what's usually done is you attempt gamma radiosurgery. This is commonly called a gamma knife, and it has this kind of freaky looking helmet device. You're locked into a stereotactic frame with this large gamma waveguide strapped to your head. Here's a picture of a person about to undergo the treatment. And what each one of these holes does is guide a weak beam of gamma radiation very precisely. The idea is that as each beam passes through brain tissue, it's too weak to cause much damage. But then at a focal point, all these beams are going to be focused on the tumor, and the level of energy that's provided at that tumor site deep in your brain is going to be high enough to damage the tissue, essentially burn out the tumor from the inside. It's very delicate, it's very scary, but it's the best approach. It's much better than physically being invasive and trying to get to the tumor from the outside. All right, so you've got brain tumors, one bad thing that can happen. What else can happen to you? You can have a seizure. And all a seizure means is you have some abnormal level of brain activity, coordinated firing or coordinated absence of firing, some persistent abnormality, either short-term or long-term. Couple of terminology bits, right? So convulsion is muscles. If you have a seizure that includes a convulsion, this is kind of the pop science movie version. You fall on the floor, you start going into spasm. Most seizures don't do that. Most seizures don't affect your motor centers and therefore don't cause convulsions. And so a partial seizure that affects only part of the brain, is mostly unlikely to cause convulsions. It may cause you to fall over, but it's not going to cause you to have that kind of spasm. If it's generalized, more likely. That means it's the whole brain. A complex seizure just means you lose consciousness. A simple seizure, and we'll talk about absent seizures in a minute as a class of that, but a simple seizure means you are conscious throughout the whole thing. Tonic and clonic, those are muscle phrases. But most seizures, if you're looking at someone who's having what's called an absent seizure, they look like this. No, the video didn't freeze out. That was me simulating an absent seizure. It's called an absent seizure because of the absence of symptoms. Effectively, what's happened is the brain went bzzz and took a moment to come back to life. Consequences? Mostly nothing. So you can have fairly low-level seizures and really not be aware of it. Repeated seizures, that's epilepsy, right? And if you have repeated seizures that are causing convulsions or that are causing loss of consciousness and so on, that's really debilitating. If you have repeated, very brief absent seizures, probably not that big a deal. If you need to treat your epilepsy, so remember HM was an epilepsy patient, had that major brain surgery. These days, what will tend to happen if you don't respond to drugs is that some electrodes will be implanted to very closely localize the source of the seizure. In general, there's going to be one point in the brain that triggers this abnormal wave of excitation. It'll be the same point every time. And so we put in these very narrow tracking electrodes to accurately localize the source of the seizure. Sometimes then what you'll do is put in basically a discharge electrode. This is a deep brain stimulation electrode. And we'll talk about those a little more when we talk about Parkinson's and so on. Effectively, what it will allow you to do is discharge, is ground out the buildup of a seizure. Generally, folks with epilepsy are able to tell more, shorter or longer periods in advance, but significantly in advance of having a seizure, hey, a seizure's coming. Often, in fact, you can train a companion animal to recognize the signs that a seizure is imminent. And so if you discharge the buildup of potential by basically zapping that brain region briefly ahead of the seizure, it's a bit like a grounding rod for lightning. You can take away that buildup of potential, you won't get the seizure. You can often control your epilepsy just by having a deep brain electrode. And the control pad, actually, the wire runs down the neck and gets implanted under the sternum usually. So you just tap your chest to activate this deep brain electrode, discharge it. If you don't have a firing disorder, so no seizures, no brain cancer, maybe you're going to have a stroke. So a cerebrovascular disorder is almost always a stroke. A stroke is any kind of event that temporarily blocks blood supply. And two major kinds, as indicated here, you can get one that causes bleeding, so basically a burst vessel in the brain, that's a cerebral hemorrhage. Or you can get one where the vessel is simply blocked, and so fuel can't get through. These are really quite common. The place where they occur is generally called an aneurysm. So that's where the blockage occurs, often causing bursting, often causing this hemorrhage, this bleeding in the brain. some more vocabulary down here. And so what happens is you get this kind of slightly delayed damage caused by apoptosis. And one of the odd things about strokes and vascular interruptions in general is often the reperfusion, the restoring of fuel supply, is more damaging than initial blockade. So what happens, and we'll show you this in a couple of slides, is you get excess neurotransmitters. When you don't have enough fuel supply, the major thing your brain is doing with fuel most of the time is maintaining neuron polarization, maintaining that membrane potential. Loss of fuel, cells start to depolarize. They lose that membrane potential. Well, you know that when cells depolarize, they fire. That's what causes action potentials. And so one of the first consequences of a stroke is that neurons start firing more than they should. That means you're getting too much glutamate being released. And glutamate in particular is damaging because it excites other neurons and then they start firing. And so you might have expected that a loss of fuel supply would cause a loss of neuronal firing. Actually, that turns out not to be the case. One of the big consequences is you get too much firing. You get a loss of ability to maintain that membrane potential. And so glutamate, as you know, causes NMDA receptors to activate, calcium comes in. If you get too much of that, so a small amount, we've looked regulated calcium entry, LTP. Too much of that, if it's out of control, generalized glutamate release, the cells have abilities to sense that. And what will happen is they will activate suicide programs. So as a consequence of this blood vessel becoming blocked, too much glutamate, then too much calcium. In order to prevent cells just from cascading, and killing all of them, the cell activates what's called apoptosis. So a series of neuronal reactions, we'll see in a slide or two, that tells the cell, hey, this is really bad and out of control. Commit suicide, kill yourself to avoid passing more glutamate on to other brain cells, other brain regions, making damage worse. So the damage from stroke is from too much glutamate being released, causing cellular death, causing cellular suicide in general. The two different types of stroke, just illustrated here, either a blockade, where fuel can't get through, or small vessels rupturing, sometimes as a consequence of blockade. Pressure builds up and poof. And then you get this intracerebral hemorrhage. Think about the likely consequences of this. As well as the loss of fuel supply, it's also going to be causing pressure. It's going to be causing a swelling effectively inside the brain. And so we'll have either temporarily or more long-term some of the effects of a brain tumor as well, because we'll get pressure damage that physically squishes other cells. I told you we'd come back to cellular suicide. The correct term for that's called apoptosis. This is again from the Greek. Too much calcium comes in, and via a whole series of cascades I really don't care about you guys remembering. Enzymes called caspases, and that you should remember. So caspases trigger this cellular suicide program. Neurons have this built-in, the ability to detect too much calcium overload. Okay, I'm going to kill myself to avoid passing on further damage to other cells. So your damage from a stroke is most likely cellular suicide. I mentioned this often happens during the reperfusion, during the resupply. When the system is coming back online, when more glucose is now available, that can actually temporarily cause even more firing, oddly enough, more calcium. And so you need to really slowly restore and gradually control the reperfusion, the resupply of fuel, so that cells are appropriately coming back, restoring the membrane potential, allowing the system to function properly, not causing further overload. Vampire bats. Cute vampire bat babies. If you're a Star Trek fan, these always make me think of Ferengi. Anyway, why vampire bats? What are they doing here? When a vampire bat wants to suck your blood, and it does, having the blood clot would be a big problem. It would impair the ability of the bat to suck that blood. And so in the saliva of the vampire bat is an enzyme called desmatoplase, which is an anti-clotting enzyme. That turns out to be one of the best treatments for stroke. And so there are now actually vampire bat farms where these vampire bats are raised, their saliva is collected, and this drug is purified out of the saliva. So if you give Desmodeplase to a person that has a thrombolytic blockage, so not a hemorrhage, because if you prevent clotting then, it's going to make things worse. But if you've got an interruption, an ischemia caused by a blockade, and you give Desmodeplase, it will very rapidly dissolve that blood clot and allow fuel supply to be restored very effectively. And you can see here, if you have an increase in the dose of Desmodeplase given within about an hour, maybe 2 hours after the onset of stroke, your chance of getting a favorable outcome is much, much higher. It goes from maybe 15% at most to more than 60% if you get this treatment within about 2 hours. Timing is really critical. If you're ever in the presence of someone, maybe grandma carrying an axe, who appears to have a stroke, get them to the ER really fast, tell them to get some anti-stroke treatment on board, and your chance of having a decent outcome is actually pretty good. Some places in the brain, you may not even notice this stroke if there's an alternate way to get the fuel. So there are some places, particularly in the brainstem, where there are alternate routes for the blood to get to a particular brain region. There are some folks who are born with a congenital abnormality where one of these blood vessels is narrowed or damaged or weak in some way. Often you may not notice that unless they become really highly stressed, get very high blood pressure, something causes extra damage. And now this ability to reroute around the blockade or weak point is overwhelmed. How do you treat? Well, it's kind of like a peripheral heart attack, right? Peripheral heart attack, that's kind of tautologist. If you have a blockade in one of the arteries leading to the heart, in general, what will happen is you put a stent. You put this wire mesh catheter and push apart the vessel to increase the ability of blood to get through. In a major brain blood vessel, such as the carotid artery, you can do something like that. It's somewhat limited because most of the vessels in the brain are simply too small. You can't get a guide wire up there. You can't make a mesh that is small enough to fit inside one of these really tiny blood vessels in the brain. So that really only applies for major arteries, major blood vessels supplying large regions of the brain. Other than that, Desmodeplase works, but the most common problem is if you've got a lifestyle and a brain situation where you're getting these blockades, it's most likely caused by bits of fat deposit somewhere else in the body being knocked off, going through the vascular system and getting to a small vessel because those vessels are so narrow in the brain, causing a blockade, causing a stroke. Some conditions, high blood pressure make things much worse, obesity make things much worse. Just sitting, being very sedentary can actually increase the risk of deep vein thromboses. Airplane flights are a well-known risk factor. And so if you're flying for a long time, get up, walk around every so often. Don't allow your blood vessels to get squished and to sit there with blood pooling and potentially accumulating a clot. All right, you haven't got brain cancer. You're not having a stroke. You don't have epilepsy. But you do have this really weird thing on MRI. What's going on in your brain? Well, in this case, I'm sorry to tell you, you've got rabies. You have an infection, not of a parasite, but of a virus. Coronavirus? No. But in fact, there are some data that coronavirus infections do seem to cause some cognitive effects. Probably in next year's version of this class, I'll have more data to talk about what coronavirus does to the brain. But rabies is very highly infectious and very transmissible within the body, right? You might remember we use modified rabies virus as a transneural tracer, as a way to get a marker between cells because it has the ability to jump across synapses. Native rabies does the same thing, but it's much more deadly. And so when you're bitten by some rabid animal and the virus gets into your bloodstream, it's going to go into your peripheral nerves, it's going to travel up the peripheral nervous system and eventually get into the brain. It does cause symptoms, just kind of like the movies. You do start producing more saliva, you do have reduced inhibitions, you want to eat flesh, you look kind of like a zombie. And by the time you get those symptoms, it's too late. You've got about 3 days to live at most. So if you're bitten by an animal and you can't trap the animal and find out whether it in fact had rabies, you get this horrible series of shots in the belly to prevent viral transmission. What you're seeing here is edema, this swelling in the brain, and it's caused by a viral infection the brain is trying to respond. You're getting some microglial response, causing perhaps an increased accumulation of fluid in this area, but it's too late. You're going to die. One of the reasons that either bacterial or viral infections can be quite dangerous is they cause what's called meningitis. So that meninges, the layers over the brain, between the brain and the skull. Meningitis is an inflammation of those layers, particularly deadly in small kids. That's why if you're a small child ever has a high fever, you've got to get them seen to make sure it isn't causing meningitis, but also, causes serious damage in adults. And again, it's because the skull isn't moving. The skull is rigid, and so swelling of these meninges will act very much like a brain tumor and compress down onto the brain. So that's one of the negative consequences of rabies. What else causes this kind of thing? Well, mercury, neurotoxins, right? You can also get infections is the wrong word, inflammation in the brain, not from something that's infecting you, but from an environmental factor. Mercury we know about because it used to be commonly used in various industrial processes. The phrase Mad Hatter comes from people who made hats and would lick the brush they were using to work with the mercury. Mercury would get into the brain, cause inflammation. So various compounds can cause brain inflammation, and it's always really nasty. You're not having a stroke. You haven't got rabies, but you did have a hamburger. Oops. Mad cow disease. So mad cow disease, bovine spongiform encephalopathy, BSE. This was something that was a really big deal when I was growing up in the UK. Back in the early 90s, late 80s, early 90s, that kind of time, we started really worrying about these folks going to the ER and having holes in their brain. So bovine spongiform encephalopathy, cow-derived, and spongiform just means has holes in encephalopathy, the brain. So it turns out this is not caused by a bacterium, it's not caused by a virus, it's not caused by any kind of DNA or RNA. This is caused by a prion. A prion, P-R-I-O-N, is an infectious protein. A guy called Stanley Krusner did some amazing work showing that the protein was the infectious element here. And when that protein is taken into the nervous system, it makes all other proteins misfold in the same way. So the prion is a misfolded form of a normal protein. It has this odd ability to make other proteins also misfold. And that causes these characteristic holes in the brain. So you can see here, if you infect a normal mouse with a prion, 12 weeks in, they've got this enormous network of holes throughout the brain. If you prevent prions from acting, nothing happens. So it really is the prion that is the infectious agent. You might notice this is actually a scabies prion. Scabies is a sheep disease. And so one of the nasty things about prions is they tend to be very similar proteins across species. And so they jump species barriers very easily. This is why mad cow disease went into humans. The way mad cow disease got started is that cows were being fed the ground up remains of other cows because it was cheap. And so they were accumulating these prions in their own nervous systems. And then when humans came along, turned cows into hamburger, ate the hamburger, now the human nervous system was also accumulating these prions. The good news is I was vegetarian at the time. So I actually kind of escaped the risk for this. We were expecting tens of thousands of deaths. It never really happened. It's not clear why, but there's now a very, very rigid system of inspection, both in the UK and in the US, for cows having any risk, any symptoms of BSE. They're excluded from the food chain really quite religiously. Another protein disease, Kuru. So Kuru is laughing sickness. The way you get Kuru is by eating someone else's brain. And so you find this amongst cannibalistic tribes, and in fact, almost exclusively amongst the young men of those tribes. And the reason for that is that the brain of your enemies is seen to be the warrior's portion. And so it's given to the most young, fit, aggressive men in the tribe. They eat their enemy's brains, and then they die. In general, don't eat brains, right? Zombies are going to die of something, but it may well be a prion disease, a disease carried by a protein. Proteins are really hard to get rid of. It's pretty easy to get rid of coronavirus. You wash your hands. it's pretty easy to degrade DNA, RNA. So viruses are fairly easy to control. Bacteria are fairly easy to control. It's actually much harder to control prions. And so CJD stands for Creutzfeldt-Jakob disease. No, I don't care that you remember that. It's the disease that BSE looks most like. So BSE was often called variant Creutzfeldt-Jakob. In both cases, these diseases cause spongiform encephalopathy, cause prion infection of the brain, leading to gross damage, gross holes everywhere. And so you can see some of the ways that was transmitted were via brain surgery instruments. If a brain surgery instrument had been used on a patient that had BSE or Creutzfeldt-Jakob and had gone through just standard sterilization that would kill off viruses and bacteria and so on, that wasn't sufficient to remove all protein traces and prions. They're really hard to degrade. And so actually there are several documented cases where you go in for brain surgery, it fixes whatever problem you had, but you end up getting BSE. Many cases where a graft of the dura from a patient who turned out to have Creutzfeldt-Jakob was put into a healthy patient, causing them to have CJD. So lots and lots of different ways that this protein could be transmitted directly into the brain of what became a new victim. Prions are not all bad. There is some pretty decent evidence that this ability of a protein to make other proteins fold in the same way may be yet another mechanism how we modify synapses. And so once you get some kind of prion form of a protein, perhaps as part of a switch from non-potentiated to potentiated. And in particular, this work was studied still in aplesia, still in these sea slugs, but at a serotonin synapse rather than a glutamate synapse. So it's one of the mechanisms that has been looked at outside glutamate NMDA channel LTP. It's possible that some proteins in the synapse may get switched into a prion form, and that may be one of the mechanisms that causes them to permanently or like semi-permanently change. So there's some evidence that we actually use prion mechanisms as a beneficial part, as a useful part of our memory processes. Proteins in general are susceptible to misfolding. So most of the time, your brain and all cells in your body, but we're going to focus on the brain, is pretty good at handling misfolded proteins. So your brain has made a protein, it's folded in the wrong way. There's a system of tagging. So an enzyme called Parkin attaches a tag. You can think of these little peach circles here as the tag. The tag is called ubiquitin. And when the cell says, oh, look, this protein is tagged, it moves it to the proteasome for breakdown. So that protein gets broken down back into amino acids. Those amino acids can then be used again. Parkin might make you think of Parkinson's, and it should. Parkinson's is one of several neurological disorders that are caused by abnormal protein folding, or in this case, a failure to remove abnormally folded proteins. So in Parkinson's, this whole Parkin system isn't working properly. And what you end up with is a buildup of misfolded proteins in the cells. So Parkinson's disease causes a loss of cells because of protein buildup. And in particular, for whatever reason, the basal ganglia are particularly susceptible to damage by protein buildup. We see this across multiple conditions. Parkinson's, the cells that are lost are primarily in the substantia nigra within the basal ganglia. This is part of the motor control system. It allows for smooth control, voluntary motions, and so on. And so Parkinson's patients have motor abnormalities. The most common is called pill rolling. And what tends to happen is the patient has this almost twitch. They keep rolling their fingers in this abnormal movement. In general, tremors at rest, some form of abnormal twitch or tick or movement are the most common symptom. It's not a cognitive effect. What happens is by the time you've lost about 90% of your dopaminergic neurons in the substantia nigra, now you see frank onset of Parkinson's. And it's caused by these abnormal, like I said, protein accumulations. Not any one particular protein, just random proteins being misfolded, building up in the cells, causing that cell basically to overload, to be unable to cope with the amount of protein debris, can't do its normal functions, eventually just dies. Several different ways we can treat that. First one of which is mentioned here, L-DOPA. So you can't just give extra dopamine. If you give extra dopamine as a drug, it doesn't cross the blood-brain barrier, can't get into the brain. That's actually the blue circles on this picture. Instead, the drug that's used is called L-DOPA. That's a precursor for dopamine. So it's a drug that can be converted into dopamine, and it can cross the blood-brain barrier. So it gets taken up by neurons, converted into dopamine, and the neurons that you still do have that haven't been killed off now release extra dopamine. You might realize, if you had time to think about this system, that's going to affect all dopamine neurons. So we're giving you a pill, it's going into the bloodstream, it's going to cause all your dopamine neurons to fire more. That may help your Parkinson's disease symptoms, the dopamine neurons controlling motor function. It's also going to affect your pleasure pathways, your addiction dopamine neurons. And so what actually happens is, this is another and very negative example of homeostasis. The increase in dopamine that's necessary to restore motor function overloads that pleasure pathway, causes it to downregulate. And so a primary side effect of L-DOPA treatment is loss of pleasure, anhedonia. It actually causes an increase in depression, an increase in suicide. So there are some really pretty major side effects because it treats all dopamine neurons, not just the ones you want to target. As a consequence of that, there's been an ongoing search for several different ways to treat Parkinson's. One example is gene therapy. So you can give a viral vector that contains the gene for making more dopamine or making more dopamine receptors, either one. Deliver it specifically into the substantia nigra dopamine neurons, and the initial trials have been fairly promising. Gene therapy in general has had a hard time, largely because it hasn't been properly regulated. There have been several instances of patients not being appropriately cared for, consent not being obtained. There was a guy I worked with when I was at Yale, and all of a sudden he wasn't there anymore. And it turned out the reason was he'd had to flee from New Zealand. He was also a New Zealand citizen because he'd been running gene therapy trials for Parkinson's without getting approval. Now, they worked, right? The patients in most cases got better, but nonetheless, the NIH, the FDA, in general, various branches of the US government didn't like this concept of unregulated, not completely approved gene therapy trials. And so he had to flee the country. I don't think he'd ever come back. His work is pretty good. So gene therapy is one approach to treating Parkinson's. You specifically give a viral vector into those cells, causing them to upregulate. It unfortunately doesn't prevent continuous damage. And so eventually you will lose all of your dopamine neurons. Deep brain stimulation is again a possibility here. And so you've got this pad under the chest bone leading up to a deep brain electrode into the substantia nigra. When you have that electrode working, it can basically compensate for the loss of dopamine function and allow you to restore basically control over your motor pathways. A third thing you can do is implant new neurons. So this is stem cell therapy. Here's the dopamine binding in the brain of a Parkinson's patient before having stem cells. And I think this is 6 months after having stem cells. And you can see those stem cells appear to have incorporated correctly. They're making more new dopamine. Honestly, I think this is the most effective treatment because it doesn't rely on kind of overstimulating the few neurons you have left. It actually provides you with more neurons on a long-term basis. DBS, though, deep brain stimulation, has some really effective outcomes. I'm going to show you a short video of one patient where fine motor control is especially important.
To Roger Fritz, the violin is not just an instrument, it's part of him, an extension of his passion for music, beauty, and excellence. Roger's mastery of the violin has put him on the world stage. Few have reached this level of expertise. But not long ago, Roger noticed something wasn't quite right when he pulled the bow across the strings.
When I would draw my bow, I suddenly had a shake in my bow. Now, for most other professions, this would not be a concern. For being a violinist, where your career depends on the stability of your appendages, this was a great concern.
Roger has what's called essential tremor, a nervous system disorder that causes shaking. Concerned, he went to Mayo Clinic where he met neurosurgeon Dr. Kendall Lee.
The amount of tremor he has will be considered pretty mild, but not in the case of a concert violinist.
Dr. Lee and Roger agreed that deep brain stimulation, or DBS, was their best chance at stopping the tremor. It involved placing electrodes into Roger's brain. But there was a challenge to overcome that.
So notice, again, the very high level of sterility. And once again, the stereotactic system, right? He's being locked into a brain frame, and they're putting this electrode very tightly, MRI guided, into a particular location.
Created a first ever situation in an operating room. You see, because Roger's tremor was so small, it would be difficult for Dr. Lee and his team to tell if the electrodes they were implanting were in the best possible place to stop the tremor.
And so what we needed a violin in the operating room.
Kevin Bennett heads Mayo Clinic's Division of Engineering. His team worked with Dr. Lee's team to create a violin Roger could play while awake during surgery.
We built and designed a 3-axis accelerometer that we could attach to the bow so that as Mr. Fish was playing, we could actually see a plot on a computer screen of the movement that really shouldn't be there.
During the procedure, Dr. Lee inserted electrodes into Roger's brain and moved them to the site where the tremor originated. Once there, electrodes fire electrical signals to hopefully stop the tremor. By measuring Roger's movements, the accelerometer told Dr. Lee if the electrodes were in the right spot. There was no guarantee it would work. Thankfully, it did work.
I thought this was a wonderful challenge to see if this technology could help in this sort of a unique situation.
I started playing right away the day I came home from Mayo. I was back playing with my colleagues with Lucid Orchestra 3 weeks later. Without the stimulator on, the tremor is quite pronounced.
You can see the difference. With the click of a controller, Roger can turn the electrodes on and off.
And that's as smooth of a bow as I can play right now.
But when it's on, Roger's mastery of the violin is absolutely clear.
I remain very, very thankful every day that I have had the opportunity to have had this surgery. And, I just do not take it for granted.
Back on the world stage to make music, beauty, and achieve excellence.
All right, it's a cute story, but it does illustrate the effectiveness, right? Deep brain stimulation is being increasingly used for all kinds of things. Weight loss, smoking cessation, the number of surgeries is really going up a lot. It's remarkably effective in many cases. Alzheimer's disease is a good example. Speaking of which, right, we're getting fairly close to the end of today's class, but here's a good example of brain atrophy in an Alzheimer's patient. So in contrast to some of the other cases where we've seen extra growth in a tumor or whatever, what you see in Alzheimer's is a loss of cells. And you can see these massively enlarged ventricles, basically because the cells are being lost from the cortex. In Alzheimer's in particular, you lose cells in hippocampus. So you lose neurons that would otherwise be involved in making and strengthening new memories. Those cells go away. And so in contrast to Parkinson's, which is a motor deficit, Alzheimer's is a cognitive deficit. Loss of memory, abnormal anxiety attacks, So you get increased amygdala activation in some cases, but memory is the most common, most obvious symptom. This is from the first paper. Dr. Alois Alzheimer drew what he saw in the brain of patients post-mortem, obviously, who had these symptoms of dementia. And what he saw and reported were these kind of shrunken, shriveled up neurons and accumulations outside the neurons of proteins, which he called plaques. We now know those plaques are made up of beta amyloid. And one thing that is commonly associated with Alzheimer's is a deficit in energy supply and use. So three things, right? Loss of cells, brain atrophy, abnormal protein effects, this might make you think of Parkinson's and so on, accumulation of beta amyloid, disruption of tau, which is a skeletal protein causing these shriveled up neurons, and hypometabolism, loss of glucose. The end result is you get these markedly increased clefts between the different brain regions, particularly down here in the temporal lobe. So here's a healthy brain, and this is a healthy brain from an 80-year-old, which looks here intact and happy. You can see in the Alzheimer's patient of the same age, there are huge losses of neurons, many, many levels of atrophy of neurons in the brain. If you look at the brain in a stain for amyloid, for the longest time, this was how you diagnose Alzheimer's, was you waited till the patient died, sliced up the brain, stained it for beta amyloid, and you see these big plaques, these big accumulations of protein. We now know that, in fact, that's not what causes the damage. These plaques are basically inert, and it appears to be that they are the consequence of the brain trying to take all this extra protein and shove it into a corner, put it in a pile, which looks like a plaque, and get it out of the way so it doesn't cause more damage. The toxic form of the protein is actually a small soluble clump of beta amyloid. And as I mentioned, the neurons themselves kind of shrink up, they get shriveled. There's a protein called tau that makes up part of the skeleton, part of the microtubules of the cell. And when that gets abnormally phosphorylated, abnormally modified, it causes the cells to get what are called tangles. They basically shrivel up. And so you have neurofibrillary tangles, these shriveled up neurons, and these plaques composed of beta amyloid protein. One way to think about that and try to remember it is you've got beta amyloid, so Baptists, or Taoists, the tau protein. And there's an ongoing controversy, which of these two is the primary cause, which comes first, maybe it's some other factor. Most of the treatments that have been tried for beta amyloid they've been tried for Alzheimer's, have targeted beta amyloid. They've mostly tried to target the plaques, and so they haven't been successful, and not surprising. There is, as yet, no good treatment for Alzheimer's disease. If you can find one, you will win a Nobel and many billions of dollars. What you see is that this tau protein doesn't just exist to cause Alzheimer's. Likewise, beta amyloid doesn't just exist to go haywire and cause Alzheimer's. Those are both components of the brain's normal response to trauma. As a consequence of that, conditions where you get repeated trauma to the brain can cause conditions that look very much like Alzheimer's disease. That might make you think immediately, for instance, of American football. And that's exactly what happens. So if you look at the brain of a 30-year-old who's been playing in the NFL for several years, it looks very much like the brain of an advanced Alzheimer's patient. You have abnormal brain loss. You have what's called tauopathy, so a degradation of cells linked to abnormal modulation of this protein tau. You have accumulation of beta amyloid. You have chronic cognitive impairment. It is certainly absolutely the case that repeated physical trauma causes those systems, beta amyloid, tau, to get activated. They're a normal part of the brain's anti-inflammatory response to a single trauma. But when it's activated repeatedly, that buildup of these proteins is what leads to the chronic long-term damage. It's one of the reasons why we now take concussion so seriously, because one concussion you'll basically recover from in general. Multiple concussions, particularly close together, are very clearly linked to long-term brain atrophy, chronic brain damage, major problems. Think about Muhammad Ali, who developed Parkinson's and couldn't speak very clearly, had long-term brain damage, died early. Boxing, unsurprisingly, is associated with this kind of traumatic brain injury. American football, likewise. Ice hockey, likewise. Anything that involves wearing large amounts of body armor and getting whacked on the head, bad idea. I coach football, soccer. Under 12 now, you're no longer allowed to head the ball because there is some data that even with a fairly soft object, the impact of heading the football repeatedly is linked to long-term, essentially old age acceleration causing dementia at an early age. So this repeated chronic traumatic encephalopathy is now a big deal. Sorry, excuse me. Beta amyloid comes from a protein in the brain called amyloid precursor protein. It gets cleaved in a certain way to cause the toxic form. I'm going to talk a whole bunch more about what beta amyloid actually does next time, because it's some of my own work, and we can look at brain insulin signaling and beta amyloid and so on. We now have the ability to diagnose Alzheimer's in a living patient because there's a stain. We can basically give a treatment that goes to the brain and lights up in the presence of beta amyloid. And you can see, control brain, very low levels. There's very little beta amyloid in the control healthy brain. But in the Alzheimer's patient brain, the whole brain is lit up. Large amounts in the frontal lobe, large amounts in the temporal lobe, closely associated with cell loss. As I said, most treatments have attempted to address beta amyloid. There have been some cases where patients get an antibody against beta amyloid that works, that appears to soak up beta amyloid in the blood and remove some of it from the brain. By far the majority, though, that doesn't work. If anything, it can cause actually more damage. So in general, patients that don't get any treatment or patients that get ineffective treatment decline pretty rapidly. So this is a 12-month study. The y-axis here is a mini mental status exam. It's a very, very simple cognitive test. Questions like, what's your name? Which state are you in? Who's the president? And so you can see a loss of 2, 4, 8 points on this test over a 12-month period. This is a pretty major loss and pretty rapid loss of cognitive ability. It can be attenuated by some of these immunization treatments, but as I said, they don't work for most people. They don't block the disease. There is no effective treatment right now for Alzheimer's disease. I alluded to this previously. One of the key factors in Alzheimer's is this disruptive metabolism, this loss of glucose supply. And you can see on this picture, it's very tightly correlated with the areas where you see extra beta amyloid being accumulated. Those are the same areas where you lose brain cells. It's the same areas where you have impaired glucose usage. So that these three things go together. It does turn out to be true that being female protects you a little bit. And the reason for that is that having more estrogen around actually modifies the way that this amyloid precursor protein gets cut. And so you end up with less of the toxic beta amyloid form. It's 1:20 in the morning, and I've been talking for more than an hour, so I'm going to call the class here. We're still talking about Alzheimer's when we start next class, and we're going to go from there to talk about the link between Alzheimer's and insulin signaling, type 2 diabetes. Turns out the biggest risk factor for developing Alzheimer's is in fact being type 2 diabetic, and that's exactly what my own lab works on. Yeah, I'm getting tired. All right, I'm going to go pause the recording and go to bed. You guys should all do the same, and I'll see you in a day or so. Good night, folks.